Pleasure to close out day one, and I get to close out by talking about something a little bit different, something we haven't really talked about today, which is structured product labeling and some strategies to improve the accuracy of SPL submissions. I'll come back to the missing LOINC part in a little bit. So here's my disclaimer slide, and I should also say that the perspective I'm bringing to this talk is a little bit different. It's not the perspective of an informaticist who actually codes or authors SPL. My perspective is that of a clinician who has worked on labeling content-related issues, be it review, development, or policy, over the last 5+ years at FDA. But really, over the course of that time, I've come to get an appreciation for the importance of SPL. It is the end labeling product for many users on many different platforms. And I've really come to believe that ideally, optimization of SPL accuracy would be a shared goal among all folks involved in labeling. The entire labeling community, and not just the informaticist who actually codes or authors the SPL. So this is how I conceptualize the labeling finish line. This is not meant to encourage anyone to run for the exits, but rather to represent, we just heard from my colleague John about some important strategies for format and appearance in labeling finalization on the way to reaching this critical milestone of getting a final approved labeling, that PDF that gets attached to the approval letter. And I echo those sentiments and think that is a critical milestone worth attaining. But once you get there, the message or my thesis is to continue to remain engaged toward the goal of submitting an accurate SPL document. So my learning objectives are to start out by reviewing downstream users of SPL. I'll talk a little bit about the accessibility of SPL, and then the importance of SPL accuracy, why it matters. From there, I'm going to dive a little bit deeper to discuss the role of logical observation identifiers, names, and codes, which fortunately is blessed with a cool acronym, LOINC, that I'll be using from here on in, to discuss the role of LOINC in SPL accuracy, and specifically why accurate assignment of sections and subsections of the prescribing information in SPL using LOINC is important. I'll identify some challenges with accurate LOINC assignment in SPL, and conclude by describing a resource that's intended to improve accuracy of LOINC assignment in SPL. So if we think of SPL as sort of the entity in the middle, who are some of the downstream users of SPL? I think many of us would think right away of healthcare practitioners, the prescribers, as well as other consumers, patients, and caregivers. But as we know, the SPL is used by other entities as well. So hospitals and pharmacies may access SPL. Educational institutions and researchers may access SPL, and importantly, healthcare information suppliers. So for example, certain drug information providers may look to mine data or extract data from specific locations in the labeling as part of the process of developing their own drug information resources or compendia. In terms of how SPL is accessed, there's a variety of platforms online through which SPL can be accessed. I'm only going to discuss a couple of them briefly here. Online labeling repositories that access SPL include DailyMed. I think many of you in the room are familiar with DailyMed. One of the newer labeling repositories is called FDA Label. It also accesses SPL. And repositories such as these share a feature in that they allow for specific searches within the SPL. So for example, again, to search for text within a specific section or subsection of the prescribing information. And I refer to this as granularity in searching. So if we look at a screenshot of the advanced search feature in DailyMed, there is indeed a drop-down menu that allows you to search within specific sections or subsections of labeling. And similarly in FDA Label, there's a search box called the Labeling Sections search box that allows you to search within specific sections or subsections of labeling. So that granular access or granular searchability of SPL is there. And that brings me back to why this issue is important. As with many things, and this is certainly true here, the quality of the search output you get from an online labeling repository is directly affected by the quality of the source data, in this case, the SPL. And I would put forward that the accuracy of SPL, for example, accurate assignment of PI sections or subsections in the SPL, can be just as important as the content of labeling. Why? Well, at least in part, because incorrect or inappropriate assignment of PI sections or subsections in SPL can impair the ability of the user to accurately search for or extract data from those sections or subsections. And I'll try to illustrate this with some concrete examples later in the talk. But before I do that, I have to take a little bit of a sidebar to describe a little bit more what LOINC are and the role that they play in SPL accuracy. As I mentioned, I am not an informaticist, so I have my LOINC 101 slide to try and demystify this concept for those of you who may not be familiar with LOINC. Essentially, LOINC codes are universal codes to identify medical terminology. And there's many different types of LOINC codes, but for the purposes of my talk, I'm focusing only on those that relate to SPL sections and subsections. They consist of 6 digits in the format shown here, and they serve to match a section or subsection name with the content of that section or subsection. So I think of this, again, in my non-informaticist way, as 2 pieces of a jigsaw puzzle. If on the one side we have the final approved labeling, say the PLR format PI as a PDF document, the hard copy, if you will, we know that in that prescribing information we'll find sections and subsections numbered and named in accordance with regulation or guidance. On the other side, you have the SPL, the electronic version of the labeling. And ideally, the LOINC code and name would serve to correspond, where appropriate, in a one-to-one fashion with those sections and subsections that are found in the PLR format labeling. To carry this concept forward specifically for human prescription drug and biological products, ideally in the SPL, we would have all those PI sections and subsections designated by PLR labeling regulations or guidances assigned to specific LOINC codes. So for example, let's look at the PLR format full prescribing information. We know that the number 6 is reserved for the adverse reaction section. Similarly, we know that the number 9.2 is reserved for the abuse subsection. So in the SPL world, there are indeed specific designated LOINC codes and names that correspond to the adverse reaction section and the abuse subsection shown there. Again, it's kind of like those 2 pieces of the puzzle fitting together. Importantly, so when a user searches an online labeling database like DailyMed for text within a PI section or subsection, say they're searching specifically within the abuse subsection, the database uses the LOINC to, if you will, decide whether that SPL contains that section or subsection. The database is not looking for the text 9.2 abuse. It's looking, is the corresponding LOINC present in that SPL? The consequence of inaccurate assignment is that the user thinks they're going to find that labeling, but potentially it could be missed if it's inaccurately assigned. So from there, any PI sections or subsections that are not designated by PLR labeling regulations or guidances would be assigned to the LOINC code for what I kind of call the catch-all LOINC code for SPL unclassified section. And there is an appropriate place and time for this LOINC code as well. So for example, let's look at our PLR format FPI again. Let's say in a specific SPL you happen to have in the dosage administration section, section 2, you have a subsection 2.4 called preparation instructions. Or perhaps in section 5, warnings and precautions, you have a subsection 5.3 called hepatotoxicity. These are not required to be named this way by regulation or guidance. So these are circumstances where use of the LOINC code for SPL unclassified would be appropriate to assign to those subsections in the SPL. Now, as I started to think about what are some of the challenges that we've seen with optimal LOINC code assignment in SPL, one of the first questions I had to address was, do all PI sections and subsections designated by PLR labeling regulations or guidance in fact have a specific LOINC code? To address this challenge a little bit, this challenge of availability of LOINC codes, it's helpful to take a brief trip back a couple of years to the last CDER Prescription Drug Labeling Conference. So this is my time machine. Some of you may have attended online or maybe participated in the room. And indeed, there was on November 2nd, 2017, an SPL session. And one of the presenters astutely presented a slide that looks something like this. I've modified it a little bit today for presentation purposes. But it illustrated that at least at that time, there were indeed several sections, subsections of the PLR format labeling that did not have a specific designated LOINC code. So as you can see in this adverse reaction section, section 6, the clinical trials experience subsection, the immunogenicity subsection, and the post-marketing experience subsection did not have a specific LOINC code. In section 8, use in specific populations, the renal and hepatic impairment subsections did not have a specific designated LOINC code. Moving further down, section 16, house supplied storage and handling, and section 17, patient counseling information, again, did not have a specific LOINC code where the LOINC name matched up with the way that the name of the section is designated to be by PLR labeling regulation. So I kind of think of this again, that puzzle analogy, it's like having some missing puzzle pieces, if you will, the missing LOINC. It's hard to have that optimal one-to-one correspondence if you don't have all of the LOINC codes there. I'll revisit this challenge a little later in my talk to discuss how it's been since addressed. But before I do that, I also wanted to discuss another challenge that we looked at with optimal LOINC code assignment. And that has to do with the uptake of newer LOINC codes. So once new LOINC codes and names are released, what does the uptake and adoption of those LOINC codes in SPL look like? To examine this, we looked at two subsections that were designated under PLLR because this was a relatively recent change. And we had a great talk earlier today about PLLR, but for the purposes of my talk, I just want to remind folks that one of the important changes that occurred with PLLR was that prior to PLLR, the subsection number 8.2 was reserved for labor and delivery. With PLLR implementation effective June 30th, 2015, that subsection number 8.2 became reserved for the lactation subsection. Similarly, prior to PLLR, 8.3 was reserved for nursing mothers. With PLLR implementation, 8.3 became reserved for females and males of reproductive potential. So the implication of this in the LOINC world is that new LOINC codes and names were developed and released for these subsections, as shown in the highlighted rows of the table. Now, in order to examine or assess how these newer LOINC codes were adopted into SPL, a search was conducted for SPL of approved NDAs and BLAs with their labeling in the PLR format. From there, we excluded those SPLs from repackers and relabelers, as well as SPLs from NDA-authorized generics. And part of the reason for this was that we previously heard that some of the issues with suboptimal LOINC code assignment in SPL came frequently from repacker, relabeler SPLs. And from there, we examined the LOINC code assignments for these subsections. So this slide presents the data for subsection 8.2, and before I present the results, I want to thank the FDA label team at the National Center for Toxicological Research, NCTR, for providing these data. And let me explain what we found. So essentially, the first column shows that of all those SPLs that met the search criteria I just described in the last slide, that in the content of the labeling had a subsection 8.2 called lactation. The second column shows what the specific corresponding LOINC code would have been for that, the optimal LOINC code. And the third column shows what was actually assigned to that subsection in the SPL. So you can see 78% of the SPL selected the most specific LOINC code for that subsection. But importantly, more than 20% selected a suboptimal or incorrect LOINC code. So 11% assigned the LOINC code for SPL unclassified, another 5% assigned the LOINC for nursing mothers, another 5% the LOINC code for labor and delivery, and 1% some other LOINC code. So the implication of this is that if a user were interested in searching for text within the lactation subsection, they would think potentially that they would get all 100% of those SPLs, but these 20% plus of those SPLs would be missed in that search. Part of the point of this exercise was to look at the uptake of newer LOINC codes, so we also examined labeling that was not in the PLLR format, that had in the content of the SPL a subsection 8.2 called labor and delivery. And with those SPLs, not surprisingly, we found notably greater fidelity in terms of accuracy of LOINC assignment. So 95% of those SPLs assigned the specific correct LOINC code for that subsection. And that just supports the hypothesis that when you release new LOINC codes and names, there is a process of uptake. It takes a while for folks to become familiar with them, the SPL authors to recognize what they correspond to, and to use them widely in SPL. I'm a visual learner, so I want to look at the data from the last slide in a slightly different way. We can also think of this data that I just presented as the current universe, if you will, of SPL containing subsection 8.2 lactation. So if a user were going in to search for subsection 8.2 lactation, they would assume that they would all kind of be housed within this one body where the optimal specific LOINC code has been assigned. But as we just saw from the data in the last slide, we have these other fireballs, celestial bodies, whatever you want to call them. You have to bear with me. I went a little far with the universe theme on this slide. But essentially what we have is we saw that there were more than 10% of the SPLs that assigned the LOINC code for SPL unclassified. Another group that, while they have a lactation subsection, assigned the LOINC code for nursing mothers. another cohort that assigned the LOINC code for labor and delivery, and a smaller subset that assigned the LOINC code for some other LOINC. And again, the point being that these other fireballs, these other celestial bodies, would be missed in a search. Now, what would the ideal universe of SPL containing a lactation subsection look like? Well, again, they would all be assigned to the most specific LOINC code shown there. We also went through this exercise or analysis for subsection 8.3, and this slide shows that we found very similar results. So of those SPLs that within the content of their labeling had a subsection 8.3 called females and males of reproductive potential, 73% of them assigned the specific LOINC code that goes with that subsection. But again, more than a quarter of them assigned a suboptimal or inaccurate LOINC code. So 16% assigned the LOINC code for SPL unclassified, 7% the LOINC code for nursing mothers, and 4% some other LOINC. And when we looked at labeling that was not in the PLLR format, that had in the content of the labeling a subsection 8.3 called nursing mothers, again, we saw that there was notably greater accuracy in terms of LOINC assignment. 95% of those SPLs selected the correct LOINC code. So again, that sort of once again supports that notion that there is a process of adoption. It takes some time for folks to become familiar with these new LOINC codes and use them widely in SPL. So we've spent a little bit of time going through a couple of challenges, the issue of missing LOINC, as well as some of the challenges I just showed with uptake of newer LOINC codes. I want to spend the rest of the time kind of trying to bring this together by talking about a document I call the LOINC document, which was developed to help with optimizing LOINC code selection. As I said, I call it the LOINC document for short. It's a PDF document. The full name is LOINC for Human Prescription Drug and Biological Product Labeling, and it was posted on the Prescription Drug Labeling Resources website recently. It's a 3-page PDF. It was developed by my team, the labeling policy team. So if you have questions pertaining to the LOINC document, you can direct them to the labeling policy team. And essentially, it contains two main components. So the first component of the document is one page that goes through some specific considerations to optimize appropriate LOINC assignment in SPL. And the second component is a series of 4 tables that correlate the LOINC codes and names with what they correspond to. So there's one table for the sections and subsections of the PLR format PI, and that table incorporates recently added LOINC codes and names that I'll share in just a moment. Because there's notable differences between the old and the PLR format, there's a separate table for the sections and subsections of the old format PI, a third table for the types of FDA-approved patient labeling shown there, and a fourth table that correlates the LOINC code and name for the principal display panel, which is a component of carton and container labeling. This is just a screenshot of part of Table 1 to give you a feel for how the table is laid out. Essentially, the first 2 columns present the LOINC code and name, the SPL world, if you will, and the third column is that attempt to bring that together with how are those sections and subsections named and as appropriate numbered by PLR labeling regulation or guidance. I mentioned earlier that back in November of 2017, there were several sections and subsections that did not have a specific LOINC code. I'm pleased to announce that some of those have since received LOINC codes and names. So now the clinical trials experience subsection has a specific LOINC code and name, as does immunogenicity and post-marketing experience. Moving further down to Section 8, the renal impairment and hepatic impairment subsections have specific LOINC codes. Section 16, house applied, storage and handling, there's a LOINC code and name currently being developed for that section. And Section 17, patient counseling information, has a specific designated LOINC code. So we encourage use of these new LOINC codes. We've also presented this information in separate forums to SPL tech folks, but we think this is a good opportunity to put this information out as well, because the thinking is if you yourself are not an SPL author, maybe you know someone at your company who is, and you can also help to disseminate this information. As I mentioned, the LOINC document begins with one page that goes through a few specific considerations, and I want to just walk through those briefly. So there's one paragraph, and I'll blow it up here. It essentially contains the overarching mantra of the document, which is to select the most specific, appropriate LOINC code to allow SPL users to identify content within each section or subsection of the PI, and to reserve use of the LOINC code for SPL unclassified section only when no appropriate LOINC code is available for that specific section or subsection. There's a couple of paragraphs devoted to the newly developed LOINC codes for renal and hepatic impairment, and they're described here. And essentially the message there is that in PLR labeling, the recommendation is to reserve those LOINC codes for when these subsections appear in Section 8, use in specific populations, and to avoid using these LOINC codes for subsections in other sections of the PI. To illustrate that a little more concretely, if we think about this partial table of contents here, fictitious example, what we mean is that if in Section 8 you have a subsection called renal impairment, go ahead and use the appropriate LOINC code and name shown there. Similarly, if you have a hepatic impairment subsection in Section 8, to use the corresponding LOINC code and name. But then if you have a couple of subsections, say in the dosage and administration section, that relate to recommended dosage in patients with renal or hepatic impairment, for those subsections, it would be appropriate to use the LOINC code for SPL unclassified. Again, that helps to sort of maintain that one-to-one correspondence where possible. There's a brief paragraph in the document about FDA-approved patient labeling, and essentially the message there is that for FDA-approved patient information, medication guides, and instructions for use, select only from one of the LOINC codes that correspond to those documents shown in Table 3 of the document, which I've shown here, and again, to avoid use of the SPL unclassified section LOINC codes. We've had challenges with that, particularly with medication guides that are receiving that LOINC code, and therefore they're being missed when folks are trying to find them in SPL. Where do you go to access the LOINC document? I mentioned earlier it was posted on the Prescription Drug Labeling Resources website. It's in the table of contents. You go to Additional Labeling Resources, and it's one of the documents under Additional Labeling Resources. You can download it as a PDF. We've come to the last challenge question of the day. Hopefully this is not Greek to you anymore. So, use of the LOINC code and name for the renal impairment subsection is appropriate for which of the following? A, any subsection in the PI that contains information on patients with renal impairment. B, the renal impairment subsection in the use in specific populations section. Or C, a subsection on dosing in patients with renal impairment in the dosage administration section. Excellent. All right, I drove that point home. So in summary, SPL accuracy can be as important as the content of labeling, and I hope I've illustrated why that can be. Accurate assignment of LOINC codes to sections and subsections of the PI is one critical component in enhancing SPL accuracy. And as we've seen, several new LOINC codes have recently become available for sections and subsections of PLR format PI, and we encourage appropriate use of these LOINC codes. With that, I'd like to thank you for your time and call John back up to the panel discussion. Thank you.